Amazing. So we will start by giving you an overview. Like Leah is going to explain the new features implemented in HALFpipe. So especially like NeuroMark. And then we will give you an update about the guidelines. So they did not change much, but we want to do like an overview again for like new people joining. And also we did a website, so it's not anymore on like Google Docs. So we want to explain anything new here. And then we want to present new tools called WonkyCon that a few people here and in Montreal have been working on to deal with multiverse analysis. And finally, we will explain like the next steps. Okay, so we can start by that. So Leah, if you want to go.
Awesome, thanks. Yeah. So we've been doing some development, especially around testing the new version of FMI 25. There's been a lot of different people testing it, which I'm really happy. So thank you to everyone. Thank you to Anthony. Thank you to everyone who's been involved with that. So that's been really cool to get some feedback on what works and what doesn't. So regarding the, okay, that's maybe I think you might need to make it full screen for the arrow keys to work. Thanks. So so one of the most requested things over the past 2 years was, can you upgrade the FMI version? And I think that's been really important to also that we keep up with the new features that they are adding with the bug fixes that they are doing that aren't being translated to the long-term support release. But the big question was, is this going to change outputs? Like, can we still call this a harmonized pipeline, even though we're like changing how the backend actually works? Violetta did some really amazing work on this. What essentially we end up doing is running a few subjects 100 times for the old version and the new version and comparing the outputs. And so she found this cool metric called the Wasserstein distance, and we did some permutation tests. And we actually see that the distributions are a little bit different, but not statistically significantly different for this plot. So you see that like the functional connectivity goes a bit up for this one parcel in the new version. But this is actually purely due to chance, according to her, most likely due to chance, according to, I'm getting messed up here with my p-value interpretation. Statisticians, please crucify me later. Okay, this is, so when we do this across the whole brain and still have now family-wise error corrected over all connectivities over roughly kind of 70,000 tests, then we see that there's a few regions where there are differences, but overall we don't have any difference. And I think this is really great to see that even though we change stuff in the backend, we've tested it, that we can kind of move ahead with FMI 25 and get all of the improvements, especially to field maps and other things from there. And performance, most importantly, it runs quite a bit faster in our testing. So that's great. But we've also added NeuroMark, which is kind of a more accurate and sensitive alternative to dual regression, which has been quite requested because it doesn't only give you spatial maps of the components that they've published, but it also gives you functional connectivity matrices. So that's kind of a very different way, more based on statistical independence kind of and multi-objective optimization to get a connectivity matrix. So that might also be interesting in case there's some concerns about using simple correlations or you have a lot of noise in your data set. So these things might be very useful there. And I think this is specifically, so Vince Calhoun's team is working with the schizophrenia working group, so they're interested in implementing that in a project. But any other projects, yeah, feel free to use it or reach out if you have any problems. I'm curious about any feedback. So all of the stuff that I'm presenting now is available in the latest development build, and we will make a release 1.3. So in a new named version that you can download and use for everything next week once we've done final testing. So, oh, no. So the other thing we've added, and this is thanks to Jurjen, who's based at University of Bonn, is multivoxel pattern analysis. This is not as interesting for resting state, but I thought I'd mention it here as well, because it's really cool. You can estimate a single brain map for every trial in your task data. And then you can use these later on to run multivoxel pattern analyses and representational similarity analyses and other things, which is really cool because that's an amazing technique, especially in the context of more naturalistic paradigms that is now also accessible for like broader ENIGMA scale studies. So I'm really happy, and yeah, thanks to Jurjen. We also had some discussion about within subject normalization, so z-scoring the image, which we've added after, so essentially in the original beta version of HALFpipe 1.0 almost 4 years ago, we didn't have this, and people told us, oh no, you need it. And now recently there's been a discussion about, oh, actually this might be giving us different results. This might actually not be good. So now it's an option, and you can enable or disable this for both FALFF and regional homogeneity. So yeah, thanks to Delin for figuring that out. And then there's another issue that's still ongoing with the debugging where field maps are sometimes not recognized. So yeah, thank you, Anthony, for pointing that out. I'm still trying to kind of replicate it on my end and figure out what is exactly was going wrong there. But I hope that we can fix that in time for 1.3 and that everything will be working in the new release. Yeah, given all of that, it will be downloadable next week, and there will be an email through the announcements mailing list. So please keep an eye out for that. And yeah, happy testing, and please open an issue on GitHub if you encounter any issues. So I'm going to hand over to Clara to give a more high-level overview.
Amazing. Thanks so much, Leah. I love the happy testing. All right. I'm going just to explain very briefly, because we already did it and there is it's on YouTube, so you can watch it. But for new people joining this call, we are doing this effort to harmonize the pre-processing and processing for fMRI, for resting state fMRI specifically right now in ENIGMA. So why do we need that? It's so if you go back to this Nature paper from 2020, it was led by Paul Drack team, I think, who asked 70 teams to run some fMRI analysis and to test some hypotheses, and 70 teams used 70 different methods. And that's the correlation of their result. So that's just an illustration of the field strongly disagree, and we need to have some harmonization on how you process your data in resting state fMRI, at least. So that's why we were doing that. That's also because at some point we want to compare results across studies within a working group, across working groups, across disease. We want to do cross-disorder analysis. We want to be able to do normative modelings. And for all of that, we need to have aligned protocols of analysis. So we conducted a survey. I extensively presented it, but we asked you, ENIGMA Consortium, and also external experts, which pre-processing step have the greatest potential to change the result, meaning which are the one we want to be very careful to harmonize. And so people agree that was compound removal and GSR, which were the steps with the greatest potential to change the result. And so then we asked for specific steps. Do you think it should never be done? Does it provide benefits, or is it required? And people tend to agree on some steps, and but also disagree, for example, on GSR, which is in purple here. Many people said it provides benefit, or even it's required, and more than a third said it should never be done. So it's one of the illustration of the steps that people strongly disagree, and we know that it's going to flip your signal. So when you look at the matrix, the connectivity matrices, you are okay, it's not going to the right direction compared to my result, and it's hard to compare after if you do not do your analysis the same way. So I presented that on this YouTube link. It's in another ENIGMA course, so if you want to know more, you can watch that. This survey and also many discussion after that led to these guidelines. So where we have common pre-processing steps where everybody agree on, there was no strong disagreement. And then we ask you to run 3 pipeline until now, which was just like COMCORE, motion parameters and scrubbing, motion parameter scrubbing, and GSR. Since that, we presented it at OHBM and many other places, and it seems like people still disagree about scrubbing. So even if it was not very much in the survey. So we decided to be very sure we will not have issues in the futures to also ask to do pipeline 2 and 3 without scrubbing, to be sure to not have like major issue in the future. So it's very easy in HALFpipe, so you do not rerun everything. You just ask like to add a specific type of denoising, but it add like a few seconds or a few minutes to do that. So it's not a big deal for you. And it will probably help us in the future, help everybody in the future. Okay, so we also try to harmonize the extracted features. So now we mainly focused on atlas-based connectivity, and with Schaeffer and Buckner for cerebellum and Christopher for subcortical. Seed-based connectivity with subcortical region, insula, and cingulate, Rio and FLFF. And based on atlas-based connectivity, we also want everybody to generate gradients. So right now we are currently implementing the gradient into HALFpipes. We will come back to that. And sorry. And so, but now, so you are like, you might be overwhelmed by those 5 pipelines. You're like, okay, so some question, like the question who came back to me many times when I presented it is like, how do we deal with that? We will get like 5 different results. We already know that. So what do I report in my paper? And what's the main results that make sense? And how can I do? So that's where Leah is going to present again.
Okay, yeah, so the kind of difficulty that came up is that, as Kyra said, it's difficult to figure out what to do. And so so we want to have some kind of magic solution that either tells us the best pipeline or tells us what is the truth across all of these results. And so we asked around a bit and got lots of different opinions on what we could do. And what came out were like 4 options, A, B, C, and D. And A is benchmarking. So we use some kind of quality metric for how good is this connectivity matrix. And we just calculate that for every site and data set. And then we choose the pipeline with the best quality metrics. Option B was calibration. So like we choose the pipeline that is best at replicating a known effect. And that's, of course, kind of difficult in functional neuroimaging because we're kind of there are quite a few results, but there's many things that have been kind of hit by the replication crisis where we are like, okay, we're actually not that sure about it, which is actually exactly why we are doing this initiative. So that's a bit harder, but there are some findings that seem very robust, especially in the context of like broader brain networks and gradients, and in terms of agent sex prediction. C, which is very popular among statisticians, is to average across models. And D would be to say, well, let's synthesize all the results in a paper and discuss them, which of course is a huge amount of work and probably will get your reviewers having nightmares. So that was kind of the situation we set out on. And then Hao Ding, who is a postdoc here in Montreal, had this amazing idea that so she did a paper comparing, I think, 10 different pipelines. And her idea was, well, let's take those methods we use in that paper and put them into a tool that can use HALFpipe outputs, can use BIDS compatible outputs, and calculate all of these metrics. I think you might need to click back into the presentation to get to the next slide. So we came up with so the ones in red are kind of quality. Well, let's start with on the left. So we ingest HALFpipe derivatives or BIDS formatted connectivity matrices, and then WonkyCon is a container that we can either put into HALFpipe or that you can run separately. and it will give you a few different metrics. So the first two up there highlighted in red are metrics of how much motion you have in your functional connectivity. So those were both come from Jonathan Power's paper from 2011. And are quite robust at detecting how much motion is in the functional connectivity. But they also heavily favor just adding more and more regressors. Like the more regressors you add, the less motion you'll have. And then there's kind of a trade-off. So, because you still want to have kind of information that might be clinically relevant, that might be relevant for individual differences in your data. So then in green are like the metrics of how good or how well like calibration metrics, how well do these matrices recapitulate desirable properties. First one is modularity, which has some problems because it can be it's a graph metric that is very sensitive to some small changes, so it might not be the best, but we're going to look into that. And the next one is the ability to predict age and sex. And in general, for sex, we see even with smaller data sets, AUCs on the order of 0.7, 0.8. So that's something that we can work with, and then probably see small differences between pipelines and use that as kind of the metric. And finally, the similarity to kind of how well we can recapitulate an average connectome, cortical gradients, and known brain networks, which might also be interesting as a metric. And then the goal is kind of to be able to get these metrics as an output from the functional connectivity estimation in HALFpipe and also from this separate tool, so that people can then either at the site directly make decisions or later on in the meta or mega analysis stage. This is an example of what comes out for our 5 pipelines. And the interesting thing is, so in blue is the baseline, which is doing no denoising at all. And on the x-axis, you see the Pearson correlation coefficient. So the higher this is, the more motion there is in the data in a way. I'm not going to go into details about the metrics. They're all in the Power paper. Yeah, what we see is that the more kind of regressions we add, if we have motion parameters, and then we do global signal regression, and we also do scrubbing, it always goes down. And yeah, ComCore is kind of comparable to baseline, but it does remove a bit. So then we can kind of see, okay, how good are these in the metric that is used here. So then we would kind of create these plots for a bunch of different metrics and enable users to build on these. Yeah, now I hand back over to Clara for some more work that comes from the team here.
Yeah, and a bunch of matrices and dataset. I guess like we are currently comparing that on like many different datasets, like Abide, HBN, many datasets from OpenFMRI to check the stability.
Exactly. The goal is to validate it before we roll it out. It's exactly. So the goal is to kind of create this as a method that we validate and then make available to people. So that means that you can start collecting data now based on the 5 pipelines. And then within the next, I mean, it's always hard to say timelines, but within the next 6 months, we would have kind of these validation results and kind of show a path forward. Because now we've established kind of these guidelines for how can we get you started, how can we get you doing multiverse, how can we get you doing all of the stuff that everyone in the field has says you need to do. But it's hard, right, because it's new. So we hope that many people will start processing their data, generating functional connectivity matrices, generating CWAS data for sites that we can put into meta-analyses. And then hopefully by the time those are collected, we will have kind of the next step ready.
Yeah. So also like we were a bit bored about these Google Docs. So we did a website thanks to Clara, Pierre, and Leah. So it's already online, so you can click on the link and you will have the instruction for like how to run things, and yeah, so it's much better and interactive. So please explore that and forget the Google Doc, because yeah, it was not amazing. Yeah, and then yeah, as I mentioned, Annabelle is just joined the team. She worked before with Lundbelek and me, and so she's going to implement the gradients and help us to test the stability of half-pipe in general. So we should have the gradient quite soon. So
Yeah, in addition, though, I want to add two questions. The first one is for Leah. I think at the very beginning you mentioned about using permutation testing to look at the consistency of results, and I was wondering about the use of interclass correlation rather than permutation testing, which is my understanding is one of the ways to, well, I mean, that's how I've at least often done the uh, testing across different ways of different methodologies for measuring the same thing.
That's an interesting idea. Actually, we haven't tried that because we kind of thought where usually you have like I don't know, like two time points for ICC, like when you retest reliability. I actually didn't consider it for this at all. So I think that's a cool idea. We should definitely compare it, and that's also good for because we're starting to write that up now to also add that in because I think that's exactly what the reviewers would ask us. So thank you. That's a really good point.
Uh-huh. Okay. And then the second question for Clara is maybe a little bit epistemic or philosophical, but so, and my question is relating to using the survey methodology for making decisions about creating a consensus approach. And I guess my concern, well, I should say early on, I was very much convinced that, like, okay, what the experts say is what we should do. But then I was thinking that the experts may be experts by virtue of them being very opinionated people. And so that the people who are, who become experts are just at that status by virtue of being opinionated. And secondarily is even if that's not the case, even if that's not the case, I think that people who answer the survey very decisively um, may be the ones that are very opinionated, and the ones that answer the survey in a more circumspect manner might be the ones that are less opinionated. Anyway, just this is maybe a rambling philosophical thing, but I think it
Oh.
should be.
uh,
Raj, you say
I
democracy doesn't work? It works great.
Democracy is horrible.
all
And
right.
I'm just I'm saying that with a caveat that there's maybe not a better system, but
Okay, so I fully agree with you first. And I got this feedback before. The survey was here to show that people disagree. So we want to use it more like as a like in the paper more like as an introduction of the problem in the field and not as an answer. So we will still show the result, but I fully agree that's not going to give you the best answer. But it still shows where people strongly disagree. But that's not how we are. That's not the only reason we are doing those we are building those guidelines. So we want to build those guidelines, like for example, those 5 pipeline, much more on those experimental testing than on the survey. And so and yeah, and it's not only the survey. We spend like a lot of time in meetings talking with many experts in the
sure.
field to get other opinion. But I still think it's very useful to see like what people want and what people think, and where people disagree. But I agree, it's not that scientific. It's more like a description of people.
Yeah. No. I appreciate the challenge of it, and I was, like I said, initially very sold on the survey, and I still value it. So thank you for explaining.
Thanks so much. Yeah, Delin?
Okay, thank you. Thank you so much for your updates about HalfPipe. It's really very exciting. I really like the new features. I have some very specific questions to Leah, especially about the normalization for the spontaneous brain activity. Yeah, we discussed about the FARFF. I understand that for FARFF, there is some kind of normalizations. So we also double check about all the other spontaneous brain activity, the real whole ARFF, to see whether there are any negative values, because they should not be negative values in these brain maps. But actually, in all of our data, the half-life pre-processed data, we found negative values. So I understand that maybe you also did normalization for this spontaneous brain activity. So I just want to know that whether this kind of normalization will change the statistical outputs, and whether we just need to clarify that, yeah, we have normalized this kind of spontaneous brain activity to make them transparency in the methods part. Yeah, that's my question. Thank you. Cody, I cannot hear you.
Yes. Okay. So by default, we normalize FALFF and ReHo within subjects, so within the image for each participant. And this was because one of the papers we referenced, the implementation of did that. And then that creates the negative values because we z-score it for a mean of 0 and a variance of 1 across all boxes. So that means essentially what you get is kind of where does this subject have a higher degree of low frequencies or Kendall W coefficient relative to other brain regions. And of course, I understand that this might now change the group results you get. And so that's why we added this option to actually be able to disable this. And I know that that's not going to help with the existing data that you've processed. So yeah, I'm sorry about any problems that were caused there. But I think it is definitely valid the way it's implemented now, and it's going to be valid if you disable it. So I think this is one of those things where we go into like all of these pipelines exist, all of these methodological choices exist, and it's really hard to kind of say this is correct, this is incorrect. It's just like all of them are going to give us different results, and now HalfPipe also lets you essentially do multiverse for this and compare kind of what is actually the impact of this methodological choice.
Okay, got it. Thank you. Thank you so much. Thank you.
Thank you for raising this issue. I think it was also very helpful to get your email about that.
Yeah, thank you.
Anthony?
Yeah, hello everyone. I just had a quick question because as most of you know, it's currently the throes of grant writing season. And so we have lots of people who are like quite eager to use our resting state data. But Enigma addiction is kind of unique in the sense that we kind of do all of our own pre-processing. So like I just calculated this morning, and we have over 2,200 subjects with resting state data, well, and with all the minimum, covariates. So that would be quite a lot to pre-process everyone, like in 6 months from now when gradients are released, or 8 months from now when another thing is released. So I'm wondering, like, is it possible to basically like use the derivatives? For example, if we selected, output a pre-processed image and did like keep all or keep some, like, would it be possible to then reuse like the derivatives rather than rerunning HalfPipe on everyone? Because then you're also using a different pre-processed image than the original output. So
yeah, I think that's exactly what we're trying to get into with the consistency check, because the idea would be, for example, you output those 5 connectivity matrices we've outlined, And then later on, for example, we add gradients, then we're also going to have a thing that will then use these existing connectivity matrices and calculate the gradients based on that. So that essentially we only require sites or people running those studies to collect those 5 matrices for every participant and only store them. And that also helps a bit with the storage issue, because I think it was also a bit of a problem to ask sites to store the preprocessed image, because that can get quite large. So that was kind of the idea with that. So we want to make sure that every future version will be kind of backwards compatible, and either it will be the same. So like what we saw earlier with the essentially completely white plot where there's like only a few connectivities that are a little bit different. Or if it is different, then we will want to use tools like WonkyCon or other quality metrics to kind of demonstrate, yeah, it gets better. So that means we can say either, well, we've improved the estimation and that's why it's different, or it's the same. And then that kind of should let's hopefully you guys move forward with kind of processing it once and then using it for all the studies. And yeah, of course, at some point we will probably need to make some changes, but I think we can keep that for the next 5 years kind of to a minimum.
Yeah, because like, I mean, once the Atlas-based stuff and the seed-based stuff is done, there wouldn't be a need to rerun those, like, once a new feature is released. But like, if the new feature requires a pre-processed image, like, if that image isn't like generated the first run of HalfPipe, then it would probably need to be regenerated, depending on, you know.
This is one of the cool new things that the fMRI developers have added with version 25, which makes it a lot faster, is that it can use, like the slowest part of fMRI prep is the normalization. So that uses ants and is like this optimization where it kind of nonlinear warps the brain and template to match. And that's the slowest part. And it does this twice, once for skull stripping and once for the actual normalization. The cool thing is that it saves this, and it saves all the motion parameters, which can also be a slow step, the motion correction for the fMRI. And then it can reapply them and recalculate the preprocessed image in only like, I think, 2 or 3 minutes. That means essentially you run it once, you save the parameters for all of the transformations, because that's in the end what preprocessing is. It's a series of spatial and temporal transformations. And because all of those parameters, all of the optimization, all of the expensive computations have already been done, it's really fast to then apply this. And so HalfPipe should already support this, but we haven't actually done extensive testing on how fast this actually goes. So the idea would be that essentially for these new features, once we roll them out and once we ask people to use them, we would then make sure that HalfPipe actually does this, and that it can then use your existing working directory and very quickly kind of regenerate the preprocessed image based on those transform parameters, and then calculate the new features without causing any problems and without kind of blowing your cluster computing budget.
Okay, so the working directory, meaning like kind of the like graphs and stuff that are built, like that's the main thing that needs
It's
to
all
get
the
saved.
derivatives folder. So essentially, as long as you keep the derivatives folder, and I guess your spec.json is always useful to keep, I think it should be fine.
Okay, so then when we're starting to run the pipeline, does it matter if you do keep all, keep none, keep some?
No, so we're going to get rid of that. So that's one of the things I'm kind of planning to work on. So I'm currently actually doing kind of an internship in Clara's group in Montreal, visiting here for a few months. So that's one of the things we want to work on, kind of getting rid of the graphs and this kind of structure we had back in the day to support the calculation of group statistics. And it's caused, I think, many problems having the graphs be pre-calculated, because it takes a long time for big data sets, right? Like you have to like run this on an interactive node, and it takes a long time to generate the graphs. It's an architectural decision that I made back in the day because it made sense, but it doesn't make sense anymore, especially for big data sets. So it's kind of, one of my priorities to kind of refactor that and get rid of it to make it simpler.
Awesome, cool, thank you.
Thank you.
Okay, so we are almost done. Also, I wanted to introduce that Paul Taylor joined the team, so he helped us, sorry, to discuss about the different metrics for the WonkyCon analysis. Do you want to just introduce yourself for us, like in 2 sentences? Sorry.
Sure. Hi, everyone. I'm Paul. I'm in the AFNI software development group, and I started chatting with Clara at OHBM, and then it spiraled into some Zoom calls and further interesting discussions. So thanks for tolerating me here.
Thanks so much. It's very useful for us.
Thank you for your amazing insights. It's really cool because I think there's a lot of experience that you can share with us, especially because we also talked about QC and how we might be able to streamline that in the future. So that's also just really cool.
Yeah, really happy to keep chatting about that in particular. So again, yeah, thanks. Appreciate it.
Okay, so I think we are done. If we don't have more point, something we can also add to the list. I mentioned it last time. I think it's like discuss about statistical issues when we have to deal with multiverse analysis. So that's on the list of the discussion we have to have in the near future. But we will have specific call about that. I don't know. Do like Frank or Paul, do you want to add anything?
No, just thank you, Cara and Leigh and the whole team for doing such a tremendous job. This is the most difficult job I've seen anyone do because not only is the method and the biology difficult, but trying to gather consensus, as I think you've done very admirably, is tricky.
Yeah, agreed. A tremendous amount of work. Even something like handling the fMRI prep update, providing the group with data to show that there's reliability in the findings. That's a tremendous amount of work, and it's a sort of unheralded. So thanks to you both.
Thank you everybody for joining this call. It's very useful to have you all. So see you on November 7, I think, for this time zone, but you are welcome to come to the other one if you are in the EU, in North America, I guess. Okay, well, have a good day.